Hello everyone, I am Dr. P. Saujanya. I welcome you all to the lecture on hypersensitivity reactions. So, hypersensitivity reactions are nothing but allergic reactions. When an allergen or antigen it infects to the host cell, it induces certain types of inflammatory responses which are known as hypersensitivity reaction. So, this is an exaggerated immune response which may lead to different forms of tissue damage. So, they are an overactive immune response and failure of appropriate recognition. In overactive immune response, it produces more damage than it prevents. An example for this is hypersensitivity reaction and graft rejection. And failure of appropriate recognition is autoimmune diseases. In this, we are discussing about hypersensitivity reaction. They are nothing but overactive immune responses to an allergen or antigen. So, hypersensitivity or allergy is an immune response results in exaggerated reactions harmful to the host. And there are four types of hypersensitivity reactions. They are type 1, type 2, type 3, and type 4. The type 1, type 2, and type 3 are antibody mediated hypersensitivity reactions, whereas type 4 is cell mediated hypersensitivity reaction. Based on the time required for sensitized host to develop clinical reactions on re-exposure. They are of immediate type and delayed type. That means based on the time required to develop the immune response to an allergen or antigen, they are immediate. That means they develop reaction immediately. And delayed type, it takes certain period of time to develop the reaction. So these two are classified based on the time required to develop the clinical reaction. Based on different mechanisms of pathogenesis, the type 1 hypersensitivity reaction is immunoglobulin E mediated. So this immunoglobulin E mediated along with mast cells are involved in type 1 reactions. And the type 2 are cytotoxic or cell stimulating reaction. Type 3 is immune complex and type 4 is cell mediated hypersensitivity reaction. These are the differences between immediate hypersensitivity and delayed hypersensitivity. Immediate hypersensitivity reactions, they appears and recedes rapidly, whereas delayed hypersensitivity reactions, they appear slowly and last for longer duration. And immediate type induced by antigens by any route, whereas delayed type are induced by antigens intradermally or skin contact. Immediate hypersensitivity is B cell or antibody mediated, whereas delayed hypersensitivity is T cell mediated. And the type 1 is also known as immediate hypersensitivity reaction. So this type 1 reaction will develop when the antigen reacts with cell-fixed antibody. The antibody involved here is IgE antibody. And this leads to the release of soluble molecules like an antigen, that is allergen, and soluble molecule mediator. The soluble molecules cause the manifestation of disease and systemic life-threatening anaphylactic shock. Anaphylactic shock is the best example for type 1 or immediate hypersensitivity reaction. And local atopic allergies, bronchial asthma, hay fever, and food allergies are the other examples for type 1 hypersensitivity reactions. The mechanism in type 1 reaction is first exposure to allergen. Here the allergen is nothing but antigen. So this allergen stimulates the formation of antibody, that is IgE type antibody. This IgE fixes by its Fc portion to mast cells and basophils. And then the second exposure to the same antigen, it bridges a bond between immunoglobulin E molecule fixed to mast cells. And this leads to the activation and degranulation of mast cells and releases the mediators. And there are the three classes of mediators derived from mast cells. They are preformed mediators stored in granules, that is histamines, and newly synthesized mediators such as leukotrienes, prostaglandins, platelets activating factors. And the cytokines produced by activated mast cells, basophils, are tumor necrosis factor, interleukin 3, 4, 5, 13, and some other chemotactic factors. These are the cytokines produced by the activated mast cells and basophils. And these mediators cause increase in smooth muscle contraction, mucus secretion, bronchial spasm, vasodilation, and increase in vascular permeability and edema. So the best example for type 1 reaction is anaphylactic reaction. So this is a systemic form of type 1 hypersensitivity reaction. Exposure to allergen to which a person is previously sensitized, like allergens like drugs such as penicillin, serum injection such as anti-diphtheric or anti-tetanic serum, anesthesia or insect venom, etc. So upon exposure to this allergen, so it releases the antibody, especially IgE antibody, and which develops the inflammatory responses such as these are the clinical manifestations produced: shock due to sudden decrease of blood pressure, respiratory distress due to bronchospasm, cyanosis, edema, urticaria. And these anaphylactic type reactions are treated by corticosteroids injection, epinephrine, antihistamines. And this is the mechanism of anaphylaxis reaction. So the IgE molecules bind to the surface receptors of mast cells and basophils. So this is the IgE antibody present on the surface of mast cells. And when the shocking dose of antigen or allergen has been present in the host, then it attaches to that of the or it binds to the IgE antibody. And this cross-linking between the adjacent antibody molecules and it increases the permeability of cells to calcium ions, and then it causes degranulation. This is the granules and they have been degranulated and they releases the inflammatory mediators. Inflammatory mediators like histamines or cytokines, interleukins, etc. With the release of these inflammatory mediators, it causes inflammation. And the next type of type 1 hypersensitivity reaction is atopy. Atopy is local form of type 1 hypersensitivity reaction. On exposure to certain allergens that induce production of specific immunoglobulin E, and the allergens involved in atopy type of reaction are inhalants such as dust, tree or pollens, mold spur, that is fungal growth. Then ingestants like milk, egg, fish, chocolate. And these are the food allergies which have been caused to some individuals. And contactants like wool, nylon, animal fur. Some individuals, they are also allergic to these types of materials. And drugs. Some individuals, they are sensitive to the drugs like penicillin, salicylates, anesthesia, insect venom, etc. And these are the different types of allergens involved in atopy type of reaction. And there is a strong familial predisposition to atopic allergy, and the predisposition is genetically determined. And these are the common diseases caused in type 1 hypersensitivity reaction. That is a systemic anaphylaxis, which is a very dangerous syndrome. And the anaphylactic drug allergy like penicillin, anaphylactic serum allergy like anti-diphtheric, anti-tetanic serum, etc. And respiratory allergic diseases like allergic asthma, acute response, chronic response, allergic rhinitis, and a gastrointestinal allergic diseases like lack of serum immunoglobulin A protein that leads to undigested protein, uh, and then allergen hydrolyse has been released, and that leads to the some of the skin allergic conditions. So the skin allergy, gastrointestinal allergic diseases, respiratory allergic diseases, and systemic anaphylaxis are the common diseases occurs in type 1 hypersensitivity reaction. Next, type 2 hypersensitivity reaction. So here the primed immunoglobulin G or immunoglobulin M, that is sensitized immunoglobulin G or M, upon the antigenic stimulus or hapten on the membrane, it causes injury and dysfunction of target cells. And this is the mechanism involved in type 2 reaction that involves cytolytic and cytotoxic reactions. So here IgG and rarely IgM antibodies has been released upon binding to that of the antigen. This is antigen. So it releases the antibodies, and this causes lysis of the cell. With the release of antibodies, it causes lysis of the cells. And this lysis of the cells was caused by the release of cytotoxic agents and cell surface antigens, and upon the absorption of the antigen on the cell surface receptor causes a disruption of normal function, and that leads to the Graves disease. So the Graves disease is the example for type 2 hypersensitivity reaction. So here in this image you can see the allergen which is present on the cell surface. It causes the release of the antibodies, and these antibodies may cause opsonic phagocytosis. Opsonin are a soluble substances which enhances the phagocytosis process. So with the release of these opsonin molecules, it causes phagocytosis of the cells. And this way it causes cell injury in type 2 hypersensitivity reaction. And the mechanism of type 2 hypersensitivity reaction are surface antigen on target cells. The target cells here are normal tissue cell, changed or modified self tissue cells, and antigens like a blood group antigen, common antigen, drug antigen, and a self antigen modified by physical factors or infection, antigen antibody complex, and antibody complement and modified self cells like these will activate the complement. These complements have the ability to lyse the target cells. And opsonic phagocytosis. Opsonin are the substances which enhances the phagocytosis. Phagocytosis is the mechanism involved in macrophages, neutrophils, etc. With this process, it causes destruction of the target cells. So stimulating or blocking effect is by promoting or suppressing the target cell functions. And this is the mechanism of type 2 hypersensitivity reaction. The antigen uh, which is present on the cell surface, it induces the production of antibodies. Here in type 2 hypersensitivity reaction, IgG and IgM antibodies has been released, and they causes activation of complement or opsonic phagocytosis, natural killer cells or phagocytosis process or stimulate or blocking. These are the mechanisms. Upon activation of the complement, it causes lysis of the target cells, and opsonic phagocytosis destroy the target cells, and by phagocytosis process it causes enhancement of antigen dependent cytotoxic cells, and all these factors will cause the target cell injury. And the stimulation or blockage, this causes change in the function of the target cells, and thereby it also causes the destruction of the target cells. Next, type 3 hypersensitivity reaction. In type 3 hypersensitivity reaction, upon antigenic stimulus, the antibody is released in the host cell and it forms a large immune complex, and this causes deposit in tissue or blood vessel walls, and that leads to the inflammation. So this is the mechanism involved here. Antigen antibody complex formation is the specific reaction formed in type 3 hypersensitivity reaction. So here you can see, upon antigenic stimulus, in the host cell, the antibodies will get stimulated and it forms antigen antibody complex. And this antigen antibody complex, it stimulates or activates the complement. This is the complement activation. With this activated complement will release certain inflammatory mediators, and these inflammatory mediators will increase the vascular permeability and infiltration with the neutrophils, and due to this increasing in vascular permeability it causes inflammation. And the examples for type 3 hypersensitivity reaction are serum sickness, which involves in organs such as skins, and Arthur's reaction. Arthur's reaction, and then kidneys that involving lupus nephritis, and lungs causes aspergillosis, and in joints it causes rheumatoid arthritis, or any other organs are involved in type 3 hypersensitivity reaction. And this reaction may be the pathogenic mechanism of diseases caused by many microorganisms. The reaction may take around 3 to 10 hours after exposure to the antigen, and it is mediated by soluble immune complex, and they are mostly of immunoglobulin G class, and although immunoglobulin M may also be involved. IgG is also involved in type 2 hypersensitivity reaction, and IgG and sometimes IgM is also involved. And the antigen may be exogenous, that is chronic bacterial, viral or parasitic infections, or endogenous, that is non-organ specific autoimmunity. And example for this is systemic lupus erythematosus. And the antigen is soluble and not attached to the organ involved. So here, whatever the antigen that has been induces the immune system or enters into the host cell, it is the soluble one, and it is not attached to any organs that are involved in the reaction. Here, polymorphonuclear cells, that is neutrophils and macrophages, they bind to immune complex via Fc portion. Fc portion is the bottom region present in antibody, and they causes phagocytose the complex. But if unable to phagocytose the immune complex, if whatever the immune complex formed here, if it is unable to phagocytose the complex, then what happens? It causes inflammation with the release of complement activations like C3A, C4A, C5A, and frustrated phagocytes. So all these causes with the release of the complements, and these complements causes lysis of the cells. So this is the example for type 3 hypersensitivity reaction, that is Arthur's reaction. So here, when the antigen enters into the host cell, it stimulates the complements. It forms then antigen antibody complex, that is immune complex has been formed. This immune complex formation will activate the complements, that is C3A, C4A, and C5A. And these activated complements will activate the polymorphonuclear cells or macrophages. And here it is activating the mast cells. And these mast cells will release the mediators, soluble mediators like histamines. And with the release of these histamines, it causes inflammation. And this is the mechanism of type 3 hypersensitivity reaction: formation of intermediate immune complex and deposition of the intermediate immune complex, and then tissue injury by the immune complex. First, initially, formation of the immune complex, and this immune complex is deposited. This deposition of immune complex leads to the release of the complement, and with the release of the complement, then it releases the soluble mediators of the complement and causes the tissue injury. These are the common diseases occurs in type 3 hypersensitivity reaction, that is local immune complex disease, Arthur's reaction, and then human local reaction, that is insulin dependent diabetes mellitus, and acute systemic immune complex diseases like serum sickness. Here the serum sickness has been formed by anti-serum, injection of the anti-serum. This induces the antibodies production in the host cell, and the antibodies along with the antigen, it causes systemic tissue injury that leads to the fever or arthritis or skin rash. And this serum in sickness has been formed by the allergic drugs like penicillin, sulfanilamide. And acute immune complex glomerulonephritis, and this is caused by streptococcus infection. And a chronic immune complex disease, it causes rheumatoid arthritis. These are the common diseases that occurs in type 3 hypersensitivity reaction. Next, type 4 hypersensitivity reaction, also known as delayed type, depending upon the time period taken for the development of the reaction, it is also known as delayed hypersensitivity reaction. Here, the interaction of primed T cells, that is the sensitized T cells and associated antigen, it leads to the infiltration of mononuclear cells, and that causes inflammatory response. Here you can see the sensitized T cells will cause the upon the contact with a specific antigen, it releases the cytokines. So the antigen, when it infects to the host cell, it induces the antibodies or T cells. Here these sensitized T cells will activates the releases the cytokines, and these cytokines affects the leukocytes, macrophages, and tissue cells. The mechanism of type 4 hypersensitivity reaction is formation of effector and memory T cells, inflammation and cytotoxicity caused by effector T cells, and inflammation and tissue injury mediated by CD4 and TH cells. CD4 is a marker present in T helper cells, and they releases cytokines and chemokines, and that leads to immune injury mainly caused by infiltration of mononuclear cells and lymphocytes. And apart from this inflammation, it may also causes cytotoxicity of CD8 cells. So here, as it is a T cell mediated, the T helper cells and the T cytotoxic cells are involved. Here the T helper cells will have the marker CD4, and T cytotoxic cells will have the marker of CD8 cells. So here the antigen or the allergen, it induces the T cells of the host cell, that is CD4 or CD8 cells. On the secondary contact, this primer T cells will release the CD4 T helper cells and T cytotoxic cells. The T helper cells releases the cytokines, interleukins, tumor necrosis factor, interferons, etc. All these factors. And the CD8, it causes directly, it kills the target cells because CD8 marker are T cytotoxic cells releases the cytokines, and these cytokines will causes the destruction of the target cells. But here T helper cells releases certain types of the factors which causes infiltration of monocytes and macrophages and proliferation of T cells and exudation and edema occurs, and that ultimately leads to the cytotoxicity. So these factors will finally lead to the inflammation characterized by infiltration of monocytes and macrophages and tissue damage. At the end, it causes the tissue damage. But immediately by the activation of CD8 or T cytotoxic cells, it causes destruction of the target cells. These are the examples of delayed hypersensitivity reactions are type 4. So the contact dermatitis, tuberculin and granuloma. Contact dermatitis will develop within 48 to 72 hours, and the clinical manifestation or appearance here is eczema, and the cells tissues involved here are lymphocytes followed by macrophages, edema of epidermis, and antigens and sites which are involved here is epidermal, that is organic chemicals, poison ivy, heavy metals. These are all the allergens or antigens involved in contact dermatitis. And the another example is tuberculin. Tuberculin is also delayed hypersensitivity reaction, and it takes around 48 to 72 hours to develop the reaction, and it develops local induration, and this involves lymphocytes, monocytes, macrophages, and the examples are intradural. This reaction is intradermal. Tuberculin and lepromin tests are examples. Next, granuloma. This takes around 21 to 28 days, and it causes hardening of the tissues, and this involves macrophages, epithelioid and giant cells, fibrosis, etc. And here the persistent antigen or foreign body presence is tuberculosis or leprosy. So the antigen tuberculin protein or leprosy protein are involved here. And the diagnostic test in vivo include delayed cutaneous reaction, that is Montague's test, patch test, examples for contact dermatitis. These are the diagnostic tests which involves type 4 hypersensitivity reaction. And the in vitro test for delayed hypersensitivity reactions include mitogenic response, lymphocytotoxicity, and interleukin 2 production. And then these reactions, type 4 hypersensitivity reactions, are treated by corticosteroids and other immunosuppressive agents. And these are the common diseases of type 4 hypersensitivity reaction. Infectious delayed type of hypersensitivity reaction, that is old tuberculin test. And contact dermatitis, here the contact epidermal agents like paints, drugs, water blister, dermatitis, etc. And this is the comparison of all 4 types of hypersensitivity reaction and the characteristics type 1, type 2, type 3 and type 4. So the antibody present in type 1 is IgE, type 2 IgG, IgM, IgG, IgM in type 3, and no antibody is involved in type 4 because it is cell mediated. Okay. And the antigen here in type 1 exogenous, type 2 cell surface, type 3 soluble, and type 4 tissues and organs. And the response time taken to develop the reaction in type 1 15 to 30 minutes, type 2 minutes to hours, type 3 3 to 8 hours, and type 4 48 to 72 hours. And the appearance of the clinical manifestation or the reaction is wheal and flare in type 1, lysis and necrosis in type 2, erythema and edema and necrosis in type 3, erythema and induration in type 4. Histology. The tissues involved or the cells involved are basophils and eosinophils in type 1, antibody and complement in type 2, complement and neutrophils in type 3, monocytes and lymphocytes in type 4. And the transformation of reaction is with antibody in type 1, antibody in type 2 and type 3. These are all antibody mediated reactions, and type 4 is cell mediated, that too T cells. And examples for type 1 are allergic asthma, hay fever, type 2, erythroblastosis fetalis, Goodpasture's nephritis, type 3, Arthur's reaction, serum sickness, farmer's lung disease, and type 4, tuberculin test poison ivy granuloma. These are the comparison of different types of hypersensitivity reactions. Thank you.